Karthi, and I'll be talking about the predictive modeling of sorghum phenotypes. This is a joint project with Purdue University, and in fact, Purdue University is providing us with all the data to do some of this stuff. And my collaborators in this work are Zhou Zhang, who is here in this room, Addie Thompson, Fang Ning, and Dr. Crawford, and Dr. Habib, Dr. Weil, and Dr. Toinstra. So this is a project funded by ARPA-E, and so yeah, I think I don't have to sell to you the need for high throughput phenotyping or why we need all that stuff, because we have been hearing about it quite a bit. But one of the things that is certainly true is that the traditional phenotyping methods are pretty time consuming, and they are manual, and they can be destructive. So I have these two images here, so that shows some of the aspects of it. So this is from Purdue University. So some of the students and postdocs and professors have set up camps, and they collect samples and they measure them using scale and all that stuff. So this is for ground truthing for our project. So obviously these kinds of manual phenotyping efforts will lead to a very increased time for breeding, and we won't be able to do this on large scale experimental panels. So keeping all this in mind, we want to do image analysis and image analytics to create better phenotyping platforms. And it has been recognized quite a bit that these days the bottleneck is not genotyping, right? I mean, genotyping has grown so much, but the bottleneck is really phenotyping. So in some sense, it becomes a very important aspect of our project to develop high throughput phenotyping platforms. And in particular, we focus on sorghum because it's an important crop for food, feed, and fuel. I think this morning we had some statistics on that. So, and it also has a natural resistance to drought and adverse growing conditions. And the other nice thing about sorghum is that it is able to tiller and regrow after harvest. So you can keep growing it through the season every time after you harvest. So, so now that we know why we need a high throughput phenotyping system, we need to know what are the requirements for this, right? So first of all, sorghum is a phenotypically diverse crop. So it can be as short as this, and it could be like 2 times my height and so on. So, and it could have different widely varying leaf counts, and it could have some of them could have panicles, some of them need not have. So this puts a lot of pressure on the phenotyping system that we want to develop. It has to be able to handle all these sorts of scenarios, and you need to be able to measure the physical and biochemical properties of the plants in all these situations. So we kind of settled on using RGB and LiDAR for looking at the geometric characteristics, and hyperspectral for looking at the biochemical characteristics. So and we want to use UAVs as the platforms for measuring all these, getting all these images, because you can use them to collect high resolution images, and they can cover large areas, which is an important thing. And we only use wheel-based sensing systems to provide some valuable augmentation for the UAVs. So in summary, our project focuses on the bigger project, in which what I'm going to present is a really small piece. It focuses on doing high throughput phenotyping in the field, which is an important distinction, using advanced sensing and analytics platforms. So these are some images, again, from Purdue University. So here is a student who is launching an UAV. And this is the Phenomobile. I think Harvey talked about it this morning. And this is an octocopter. And these are some of the cameras that we were planning to use. So and all of them, many of these imaging systems can be mounted in one of the sensor platforms. So one of the things I realized, I mean, my background is machine learning, electrical engineering. So one of the things I realized when I got into this project is that you need to really know how things happen in the field. So it's not sufficient that you sit with your data and you do all your analytics. It's not sufficient. So and Addy has been very kind, in telling us a lot of things about how things happen in the field. And this is one thing that is important to know, how the field is set up, right? How the experiment is set up in the field. So in this particular experiment, we are using this so-called calibration panel, where we have totally 18 varieties of sorghum that are planted in 6 replicates. 4 of the replicates are planted in high density, which is 223,000 plants per hectare. And 4 of the replicates are in low density. And the varieties are randomized across each column, which we call as the range. So this is how the field was set up, and this is the field from which we got the data for our analysis and experiments. So as we know, we want to predict the phenotypes. So the phenotypes that we want to predict are first the height, then second one is the diameter of the stalk, and the third one is the volume. So the height and the diameter values were collected by hand from 3 representative plants in each of the plots. So there were 144 plots. So and they were collected at several time points. So we analyzed 3 time points. The stalk volume was approximated from stalk diameter and height, considering the plant to be like a cone. So the volume is pi r square h by 3. And the data was, so totally there are 144 plots, but we used only 108 plots because we couldn't go into 36 plots and collect the data. They were so dense. So we had to use only 36 low density plots. And we used 2 different dates, August 6, 2000, I'm sorry, this should be 2015. August 6, 2015 and August 21, 2015 for this analysis. So this is the ground reference data. This is the ground truth data. So we want to use the image data to predict this, right? So what is the image data that we use? So we use 2 types of imaging modalities. The first one is hyperspectral imaging, and the second one is RGB. So first I will talk about the hyperspectral images. They were acquired from UAVs flying at 16 meters per second at an altitude, fairly high altitude of 120 meters. So this corresponds to a ground sampling distance of 5 centimeters. So you have 5 centimeter pixels. And the raw data had 278 bands with 2.2 nanometer resolution. So this is an example of the hyperspectral data cube. So imagine it like a 3-dimensional data, right? So the first 2 dimensions are the spatial dimensions, and the third dimension is the spectral dimension, right? So then we pre-processed it or post-processed it to remove some of the bands and also average the bands to finally have 114 dimensional pixel. And we used a feature called extended multi-attribute profile. Basically, it is a morphological feature, so it can be applied on images, but we got 2 principal components out of these hyperspectral data, and we used these morphological profiles on 2 of them to get and stack the features. So that's how we got this EMAP feature for hyperspectral data. And there are also these hyperspectral indices that people have used quite a bit, and it's very popular in the I think in the phenomics community also. So some of the features that we used are, first of all, they convey different things about the different aspects of the vegetation. For example, some of them can tell you about the chlorophyll content, some of them can tell you about the plant senescence, some of them can tell you about the water stress, and so on. So they are pretty useful, and I mean, these have come out of lots of years of research, so there is no reason why we shouldn't use them. So we considered 5 different indices that we thought were pretty useful. So so some of the indices that we used were simple ratio indices, and also the popular normalized difference vegetation indices. So this is the data that we had. So we had, from hyperspectral, we had 3 types of data. So one is the mean spectrum itself, the other one is the attribute profiles, and the third one is the hyperspectral indices. So from the RGB data, what we did was Professor Habib's group, they analyzed the RGB data and they provided us with point clouds. So you can think of it like a substitute for LiDAR data, right? So LiDAR data gives you the 3D point clouds directly, but you would be able to get the 3D point clouds in some sense just using RGB data and doing photogrammetric reconstruction. So you can create a 3D point cloud. So these were acquired from UAV flying at 8 meters per second at a relatively lower altitude compared to the hyperspectral. So at 15 meters, so it has a better resolution. So it's 1.5 centimeters. And as I said, this is the second step is the one that is used to create the 3D point clouds. So you have images from various flight lines, and you can, kind of stitch them together and also infer the depth. So that is called bundle adjustment. So you finally get a 3D point cloud. So once you get a 3D point cloud for each plot, one of the features that have been very useful, in these communities is the height features. So you can construct height histograms from the 3D point clouds, and the height histogram kind of looks like this, for example. So so these are the two different height histograms for two different varieties of sorghum. So the first one is a grain sorghum, and the second is a forage sorghum. So the blue one is the low density plot, and the red is the high density plot. Just look at how different the height histograms are across the two plots, right? Particularly in the forage sorghum, which is like a very bushy structure. So now towards something on exploratory analysis, so we fit individual features to phenotypes. So we see that some features correlate well with both the high density and low density plots, but some features correlate better with high density plots compared to the low density. So and we also performed some predictive modeling to actually predict the phenotypes. So we ended up using supported regression because we figured that gave us the best performance in terms of everything. And we also figured that when you fuse multiple features, we got a much better performance rather than using a single feature. And this could be performed very easily by stacking the features and normalizing them. So these are some of the predictions. So one thing we figured was, if you use a systematic partition, if you try to predict the high density using the low density or low density using the high density, the performance was not as good because, one model does not fit all, certainly. So we had to randomize it to get a better performance. So one of the things we are exploring is how to, get these different models, but also share some information between these two models, the high density and low density. These are results for two other dates using features on 8.4 and predicting phenotypes on 8.21, and features on 8.4 and predicting phenotypes on 8.6. So the results are similar, but as you go back in time, your predictivity kind of decreases. So we also want to explore, time series based analysis where you can bundle all these time points and create better predictions. So to conclude, we performed both exploratory analysis as well as predictive modeling. And one of the things we figured is that simple stacking is not sufficient, and we want to explore better ways to fuse the features from the different modalities. The other observation is that one model fits all doesn't work, so we have to develop models that share information. And we also want to investigate the aspect of using multiple time points. So some acknowledgements, first to ARPA, next to the amazing Purdue phenotyping team who actually go in the field and collect all the data and do all the hard work, and to the IBM Terra project team for all the great discussions, help, and motivation. And these are some references.